Hello guys, welcome to my second lecture on diagnostic serum enzymes. So today we are going to talk about CPK enzymes or creatine kinase enzymes. So creatine kinase, to begin with, this is basically a dimeric enzyme, and there are three different isoenzymes of creatine kinase. Isoenzymes are those enzymes which differ in amino acid sequence but perform the same function. So these enzymes are those enzymes which are different isoenzymes. So this is the definition, which are different in amino acid sequence, okay, but essentially they perform the same function. So these are isoenzymes, and there are three different isoenzymes available of creatine kinase. So the first one is CK1 or CKBB, which is mostly found in the brain and smooth muscle. The second one is CK2 or CKMB, which is mainly found in the heart. And the third one is CK3 or CKMM, which is mainly found in the skeletal muscles and the heart. So these three are very important. So sometimes questions are being asked about the locations of finding of the three different creatine kinases. Next, if we talk about assay means how will we qualitatively find out whether creatine kinase is being formed within a system or not. So as you can see here, the substrate for creatine kinase is called phosphocreatine or creatine phosphate. So this is the substrate, and with the help of creatine kinase, phosphocreatine is converted to creatine. Okay, so phosphocreatine is converted to creatine with the help of creatine kinase, and this is the reaction which is catalyzed by creatine kinase. So phosphocreatine is being converted to creatine. So at that same time, when phosphocreatine is being converted to creatine, it is utilizing ADP and it is generating ATP. Okay, so this is a kinase enzyme. Kinase means which will generate ATP. So this enzyme generates ATP, and what we do with this ATP, we use it in glycolysis step. So this ATP is nothing but it is used in glycolysis. Where it is used in glycolysis, you remember the first step of glycolysis is from glucose by the action of the hexokinase enzyme. This is another hexokinase enzyme, and with the help of ATP, okay, you give glucose 6-phosphate. Okay, then from glucose 6-phosphate you generate fructose 6-phosphate, and ultimately you generate, after a series of steps, you generate pyruvate. Okay, so this is the ultimate step of glycolysis. Now there is one intermediate step where NADPH is produced. NADP is reduced, and NADPH is produced. Okay, so there is one step within glycolysis, and as I have talked about in my previous lecture, so you can measure this NADPH at 340 nanometer wavelength. Okay, so why 340 nanometer? Because this is the wavelength at which maximum absorption occurs. So if NADP, now we will backtrack. So if NADPH is produced, that means ATP has been utilized by hexokinase, and from where this ATP came, this ATP came from this reaction. That means this reaction has occurred. Phosphocreatine has been converted to creatine. That means what? Creatine kinase is present. Okay, so this is the bioassay technique that we use to quantify creatine kinase. This is again another reaction which is showing that ATP that you are generating from the previous reaction of creatine kinase, you are using it to in the glycolysis step.